Hello students, we are here again to discuss a sequence of life processes. As you last time did digestive system. Today we will be talking about certain general aspects of life processes and mainly nutrition in plants. So let's begin without wasting time. Life. What is life? Living beings can move, living beings can locomote, living beings can breathe, they can excrete, they can reproduce. But sum total of metabolic processes going inside the cell is what defines life. And out of all the metabolic features, these are the biochemical reactions. And you as very well know, biochemical reactions are of two types: catabolism, breaking up reactions; anabolism, building up reactions. Till these reactions are going on inside the cell, we are living. The moment these reactions stop, life is no more. And for that, all the processes are governed by these biochemical reactions only. Living things move, respond to stimuli. This is the most defining feature of life, which in higher biology you will be talking as consciousness. They grow, they reproduce, they need nutrients and compatibility of self-repair. Move तो गाड़ी भी कर सकती है यार, ग्रो तो माउंटेन भी करेगा, डस्ट पार्टिकल्स ऊपर आ जाते हैं. इसको कहते हैं ग्रोथ फ्रॉम आउटसाइड. लिविंग बीइंग्स ग्रो फ्रॉम इनसाइड. All living beings don't reproduce. Mule doesn't reproduce. Infertile couple don't reproduce. Agreed, they are the characteristic feature, but the defining feature is this plus metabolism. And to carry out all these, we need nutrients in the form of energy. However, viruses are neither living nor non-living. As you know these days about coronavirus, when it enters the body, enters the lungs, enters the human cells, only then it becomes living, because it is using cell's own machinery to replicate, to duplicate itself. Otherwise still it is outside the cell. Viruses are considered to be dead, otherwise they become living. There are some basic processes which are known as life processes. Nutrition, taking food; metabolism, I have already explained; reproduction, excretion, respiration, transportation, or circulation, we can say, in animals. All these life processes are governed by the metabolic reactions. Now, as we said, they reproduce, they move, nutrition is there, excretion is there, respiration is there, growth is there, but the defining feature is consciousness. Whether it is bacteria, whether it is amoeba, whether it is weed, whether it is plant, whether it is fungi, we all respond to stimuli. This is what is known as consciousness, my dear friends. Sum total of chemical reactions, as I told you, constitute a living organism. They are anabolic reactions, which are building up reactions. Catabolic reactions, braking reactions. For example, respiration. It always releases energy, and here the requirement of energy is there. For example, photosynthesis, making proteins from amino acids. These are anabolic processes. Now we come to nutrition. Nutrition means to obtain nourishment, to obtain food, to obtain nutrients by organisms. This food can be prepared on its own or can be obtained from others. Plants are autotrophs, animals are heterotrophs. So, plants, blue-green algae, cyanobacteria, algae, bryophytes, pteridophytes, they are all autotrophs. Heterotrophs, animals. In this we also include saprotrophs, fungi. We also include parasites. Meaning, in heterotrophs, the living being will not produce its own food. It will obtain food from some or the other source. So it's very clearly given. Animals, fungi and many bacteria are heterotrophs. Whereas plants, algae, bacteria like cyanobacteria, they are autotrophs. Now plants can be of two types. Photoautotrophs, which will produce food in the presence of sunlight. Chemoautotrophs, which will use chemical energy. And they don't require any sunlight to produce food. Phototrophs, they trap solar energy and use it for synthesis of food, and carbon dioxide, water are required in this process. Chlorophyll is the main molecule which traps energy to synthesize glucose. Some bacteria like sulfur bacteria, nitrifying bacteria, capture energy released during chemical reaction and use it to synthesize food. Actually, carbon dioxide needs source of hydrogen to produce glucose. It can be from H2S, it can be from acetic acid. So, chemicals are involved. So water is not required for photosynthesis, because photosynthesis, another name is carbon dioxide fixation or autotrophism. Another term used is carbon dioxide reduction. So they are using chemical energy to synthesize their food. Now types of nutrition, mainly the heterotrophic nutrition, parasitism, endoectoparasites, saprotrophs, mainly fungi, holozones, mainly animals, including human beings, including single cell amoeba. They are holozones. They take their entire food. Saprotrophic nutrition, you can see the bread mold growing on the bread. Rhizopus, mucor. They digest outside their body. That's why the digestion is extracellular. They grow on dead decaying organic matter. That's why they are known as saprotrophs. The characteristic feature of fungi is they are saprophytes or saprotrophs. Parasitic nutrition, we can have ectoparasites, We can have endoparasites. Now ectoparasites like Scuta, Amarbale, what it does, it produces its roots known as hostras in xylem and phloem components of the plant, and themselves don't prepare any food. That's why they are total stem parasites. Whereas endoparasite, Plasmodium vivax, which causes malaria, which has become a household name. So as you go ahead in biology, there will be 4 species of Plasmodium: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, which causes reoccurring fever, where chills are there, body weakness will be there, anemia will be there. And Plasmodium will invade first the liver cells, then the RBCs, destroying RBCs, causing anemia and weakness. So this causes malaria. This is an endoparasite within the body. Parasite never wants to destroy the host. It takes nutrition from the host to the extent that the host is not killed. That you should remember. Then we have holozoans, mean feeding on solid food particles. They take complex organic food and convert it into simple molecules by digestion. For example, human beings. We take in food and digest it in our alimentary canal and finally absorb it. Same as amoeba, it engulfs the food. In the food vacuole, intracellular digestion occurs. It is also holozoan. Nutrition in plants, this is where we want to talk, because the lecture on digestive system has been already delivered. You just can watch it. That has been done in details. But now we come to plants. Green plants are autotrophs. They require carbon dioxide, water, sunlight, chlorophyll to manufacture glucose and liberate oxygen. This whole process is known as photosynthesis, and the equation for photosynthesis is summarized here. Six carbon dioxide plus 12 water gives glucose plus 6 water plus 6 oxygen. So they are converting light energy into chemical energy, where glucose is formed, the bonds are formed. That is what is photosynthesis, and as I already told you, photosynthesis is carbon dioxide reduction. In the course of your lectures, you will find carbon dioxide plus water gives glucose plus oxygen. Initially, we used to think, if I break up this glucose, if you allow me to write it like this, it seems that carbon and water are combining and the oxygen which is released is coming from carbon dioxide. This was wrong. Actually, the oxygen which is released is coming from water by photolysis of water. Just like in chemistry you talk about electrolysis of water, same way photolysis of water will occur, and the oxygen which is coming from water rather than carbon dioxide. So it is carbon dioxide fixation or carbon dioxide reduction. Water is only being used as a source for hydrogen to reduce carbon dioxide into glucose. So now, process by which green plants make their own food from carbon dioxide and water by using sunlight in the presence of chlorophyll is called photosynthesis. During this process, light energy is converted into chemical energy. This is a section of leaf, of a dicot leaf. This is the upper epidermis, cuticle, which is the waxy layer which will reflect sunlight. Lower epidermis, where are present the stomata along with guard cells, through which gaseous exchange will occur. The upper layer is palisade parenchyma, which has chloroplasts and will mainly be responsible for photosynthesis. Then spongy parenchyma, which have large intercellular spaces so that gases can diffuse in and out very easily. Then bundle sheath is there, xylem towards the upper side, phloem towards the lower side. It's a basic diagram of a leaf. Whatever is present between the two epidermis we call it as mesophyll. So in a dicot leaf, mesophyll can be divided into palisade parenchyma and spongy parenchyma. Now let's look at the structure of chloroplast present in the leaf cells, palisade parenchyma. Chloroplast consists of a double layer structure with outer and inner membrane. Then there are present stack-like structures known as thylakoids. These. Together they are known as granum. Plural is grana. As you know, chloroplasts are semi-autonomous cell organelles because they possess their own DNA, their own ribosomes, although they are dependent upon cell for some nutrients, for some enzymes, but they have the power to divide on their own. That is why we call it as semi-autonomous. Whatever is left between these thylakoids, this region, is stroma. As we proceed further, these thylakoids will contain chlorophyll molecules. And I will be dividing photosynthesis into two processes, light reactions and dark reactions. Light reactions will occur on the grana and dark reactions will occur in the stroma. For that, just give me 2 minutes more. Sunlight is required for photosynthesis. Water, chlorophyll, carbon dioxide. Let's have some experiments for it. What we do is, we always take a potted plant and keep it for 24 to 48 hours in darkness. This is the first step. The basic aim is to de-starch it. कि जो स्टार्च उसने ऑलरेडी स्टोर कर रखी है वो यूज हो जाए इन द प्रोसेस ऑफ रेस्पिरेशन एंड अदर प्रोसेसेस ऑफ द प्लांट, ताकि एक तरह से उसे हमने स्टार्व कर दिया. दिस इज द फर्स्ट स्टेप. सेकेंड स्टेप, वी एक्सपोज इट टू सनलाइट. Then we remove the leaf in the evening, and this leaf is heated in a water bath, dipped in alcohol. Alcohol is organic solvent, वो सारी chlorophyll खींच लेगी. As a result, leaf will get bleached. अब जब आयोडीन टेस्ट लगाएंगे तो ये ब्लीच्ड लीफ विल टर्न इनटू ब्लू ब्लैक. ये बेसिक स्टेप्स हैं. तो अब एक एक करके एक्सपेरिमेंट देख लेते हैं. द फर्स्ट स्टेप इज सनलाइट इज नेसेसरी फॉर फोटोसिंथेसिस. It is the ultimate source of energy and rate of photosynthesis maximum in red light. अब अगर हम क्या करें, एक पॉटेड प्लांट लें उसकी लीफ को अगर हम ब्लैक पेपर से कवर कर दें, ये पोर्शन will never synthesize starch, indicating sunlight is necessary for photosynthesis. Chlorophyll is necessary for photosynthesis. ये chlorophyll molecule है जिसमें मैग्नीशियम बीच में है, ये पायरॉल रिंग्स हैं, ये आप हायर क्लासेस में पढ़ोगे। तो these are light trapping centers. अगर हम कोई ऐसी लीफ लेते हैं जिसमें नॉन ग्रीन पार्ट हो, जैसे कि ये देखो, ये वेरीगेटेड लीफ है जिसमें येलो पोर्शन है और ग्रीन पोर्शन है। जब इसको सनलाइट पे एक्सपोज किया, फिर इसको ब्लीच किया वाटर बाथ में अल्कोहल के साथ, आयोडीन टेस्ट परफॉर्म किया, तो सिर्फ ग्रीन पार्ट जो है वो ब्लू ब्लैक हुआ, येलो पार्ट कभी भी ब्लू ब्लैक नहीं हुआ। इसका मतलब that only green part which contains chlorophyll will produce starch, never any other. और ऐसे लीफ को वेरीगेटेड लीफ कहते हैं, पैड लीफ, जैसे कि क्रॉटन लीफ है जो ऑर्नामेंटल प्लांट है। Carbon dioxide is necessary for photosynthesis and for respiratory exchanges. The stomata along with guard cells, they are responsible for exchange of gases. These are minute pores present on the surface of leaf. They will help in respiration, they will help in gaseous exchange during photosynthesis, they will help in transpiration also. Now to prove that carbon dioxide is necessary for photosynthesis, हम क्या experiment लगाते हैं, दो बेल जार लेते हैं, एक में QH रख दिया, इन दोनों प्लांट्स को सनलाइट में एक्सपोज कर दिया। जब इसकी लीव्स को ब्लीच किया और आयोडीन टेस्ट लगाया तो नथिंग हैपेंड, नो ब्लू ब्लैक। इस केस में द होल लीफ बिकम्स ब्लू ब्लैक। तो हुआ क्या, इस KOH ने कार्बन डाइऑक्साइड को एब्जॉर्ब कर लिया टू फॉर्म पोटेशियम बाइकार्बोनेट, तो इसलिए इस प्लांट को कार्बन डाइऑक्साइड मिल ही नहीं पाई। दैट इज वाई कार्बन डाइऑक्साइड इज एसेंशियल फॉर फोटोसिंथेसिस। Water is used in photosynthesis taken up from soil by roots in terrestrial plants. Other materials like nitrogen, phosphorus, iron and magnesium are taken up from the soil. Nitrogen is an essential element used in synthesis of proteins and other components. Even chlorophyll में भी ये जो pyrrole rings हैं इनमें नाइट्रोजन है, अमीनो एसिड्स में नाइट्रोजन है। तो जैसे आप मैक्रोन्यूट्रिएंट्स की बात करते हो, कार्बन, हाइड्रोजन, ऑक्सीजन, नाइट्रोजन, आई ऑलवेज मेक स्टूडेंट लर्न कॉर्न, पस, पका, मज, कार्बन, हाइड्रोजन, ऑक्सीजन, नाइट्रोजन, फॉस्फोरस, सल्फर, पोटेशियम, कैल्शियम, मैग्नीशियम, दे आर द मैक्रोन्यूट्रिएंट्स। which are required in higher quantities. Corn, pus, paka, maj, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium और magnesium। इन तीनों सब में से भी NPK, nitrogen, phosphorus, potassium are very very important. The steps include absorption of light by chlorophyll, conversion of light energy to chemical energy and splitting of water into hydrogen and oxygen. This is known as photolysis. Reduction of carbon dioxide to carbohydrates. So as I have been saying that water is just a source for hydrogen. These are the three main steps that you have to learn. Absorption of light by chlorophyll, conversion of light energy to chemical energy and splitting of water molecule into hydrogen and oxygen, reduction of carbon dioxide to carbohydrates. As I said, light reactions occur on the thylakoid, that is the granum part. The main aim of light reactions is to produce high energy molecules like ATP and NADPH. Then in the stroma part, dark reactions goes on, which are biosynthetic reactions, in which C5 compound will combine with carbon dioxide to form two C3 compounds, which will further fuse to form glucose. तो ये Kelvin cycle आप higher classes में पढ़ेंगे. But for all these dark reactions to go on, ATP and NADPH are required, and these have been synthesized in the light reactions. This you have to remember. Now, it is a self-sufficient process. Light reactions ATP, NADPH बना रही हैं जो कि यूज हो रही हैं डार्क रिएक्शन में. बट डोंट मिस्टेक मी, डार्क रिएक्शंस का मतलब नहीं है ये कि डार्क में ही चलती हैं. डार्क रिएक्शंस कैन गो ऑन इन लाइट एज वेल एज डार्क, बट दे आर इंडिपेंडेंट ऑफ लाइट, इसलिए इनको डार्क रिएक्शंस कह दिया. दे आर बायोसिंथेटिक रिएक्शन जिसमें ग्लूकोज बनेगा. दिस इज हाउ दे आर शोइंग that how water is splitting in the presence of sunlight to form hydrogen ions. They will be taken up by NADPH. These electrons will jump from one center to another to produce ATP, and oxygen is reduced. Formation of similatory pairs or reducing pairs, that is ATP and NADPH2, are used in dark reactions. For formation of one molecule of glucose, you require 18 ATP and 12 NADPH. But for the time being, this is beyond your levels. It is only when we talk about C3, C4 cycle, Kelvin-Benson cycle, or other cycles that you'll come to know that where ATP are used, where NADPH are used. Dark reactions are Blackman's reaction, biosynthetic pathway, that is they will synthesize glucose. It's very clear they are light independent reactions. And formation of glucose occurs. The dark reactions occur in stroma. They use ATP and NADPH produced during light reaction. Plants capture carbon dioxide from the atmosphere, which combines with the organic molecules to form C3 compound. Chemical energy stored in ATP and NADPH is transferred to this C3 compound for production of glucose through Kelvin cycle. तो जैसे मैंने बोला कि Kelvin cycle is beyond your scope right now, but C3 join कर जाएगा दो to form your glucose molecule. तो इसके लिए हमें टाइम मिला तो मैं आपको पहले ग्लाइकोलिसिस कराऊंगा, फिर अगर कैल्विन साइकिल सीखना होगा आपने तो रिवर्सल ऑफ ग्लाइकोलिसिस पहले पढ़ना पड़ेगा, तब कैल्विन साइकिल आपको समझ आएगा. बट अभी सिर्फ आप ये जानिए कि कैल्विन साइकिल जो है, कैल्विन बेंसन साइकिल, दैट इज़ अ बायोसिंथेटिक साइकिल. डार्क रिएक्शंस हो रही हैं उधर कोई दो C3 कंपाउंड्स मिलके C6 ग्लूकोज बना रहे हैं. नाउ दिस इज योर लेवल. Dark reactions occur in, right answer will be stroma. Oxygen is liberated in photosynthesis from water, photolysis. थोड़ा सा बता देता हूं. हमारे पास water breaks into hydrogen ion plus OH negative ion. Another water breaks into hydrogen ion plus OH negative ion. Another water breaks into hydrogen ion plus OH negative ion. Another water breaks into hydrogen ion plus OH negative ion. ये OH negative ion अपने इलेक्ट्रॉन दे देते हैं to become OH neutral. इनमें से ये वाटर रिफॉर्म हो जाता है, और ये मॉलिक्यूल्स 2 ऑक्सीजन के उड़ जाते हैं. These hydrogen ions are picked up by NADP for reducing carbon dioxide. और जब इलेक्ट्रॉन्स जंप करते हैं फ्रॉम वन फोटोसिस्टम टू अनदर, तो रास्ते में ATP बनते हैं. तो जब तक आप थोड़ा डिटेल्स नहीं कर लेते, थोड़ा सा इतना जानिए कि इलेक्ट्रॉन जंप फ्रॉम वन रिएक्शन सेंटर टू अनदर और ATP प्रोड्यूस होते. Chlorophyll contains which ion? Magnesium. मैंने बोला magnesium is joined by 4 pyrrole rings, ऐसे. फिर एक phytol chain भी होती है, ये chlorophyll है. How many ATP are used in synthesis of one molecule of glucose? 18. मैंने पहले ही बोला. How many NADP? H 12. For photosynthesis, carbon dioxide requires water, source of hydrogen, hydrogen sulfide, copper. As I said, it is carbon dioxide reduction, so it requires source of hydrogen only. वो तो in the course of evolution, जब पहले Chemoautotrophs थे, जैसे सल्फर स्प्रिंग बैक्टीरिया की बात करें, वो हाइड्रोजन सल्फाइड से हाइड्रोजन ले जा रहे हैं. नाइट्रिक एसिड बैक्टीरिया की बात करें, वो नाइट्रिक एसिड से हाइड्रोजन ले जा रहे हैं. ऑर्गेनोकीमोट्रॉफ्स की बात करें तो वो एस्टिक एसिड जैसे एसिड से हाइड्रोजन लिए जा रहे हैं. तो ज़रूरी नहीं है पानी चाहिए. हाँ, इन द कोर्स ऑफ इवोल्यूशन, बिकॉज वाटर वॉज मोर अबंडेंट, तो इट वॉज यूज्ड एज यूनिवर्सल रिड्यूसिंग एजेंट विच यूज्ड टू गिव हाइड्रोजन to carbon dioxide for its fixation or reduction.